Dr. Lindsay Burwell has been Assistant Professor of Chemistry at Wells since April 2015. I'm pleased to share that she's recently been approved for tenure and will be promoted to Associate Professor in just a few weeks. Congratulations. Dr. Burwell is a member of the Education Policy Committee and Chair of the Health Science Program. Dr. Burwell earned her BA in Biology at SUNY Potsdam and her PhD in Biochemistry and Molecular Biology at the University of Rochester. Dr. Burwell is interested in investigating the chemical reactions that drive metabolism and control mitochondrial physiology. This underlying focus has driven her research in many areas of the biomedical sciences, from cardiac ischemia, reperfusion injury, and drug development, to mitochondrial protein assembly, to identifying biomarkers associated with weight gain and metabolic stress. Dr. Burwell has mentored student research projects at Wells College that align both with her research focus and with student interest. Her students have presented their research results at the National Conference of Undergraduate Research, NCUR, the National American Chemical Society Conference, Undergraduate Research Day at the University of Rochester, and the Rochester Academy of Sciences annual meeting. I am delighted to introduce Dr. Burwell to you, and I think her presentation tonight is going to be both fascinating and fun. It is titled Accidental Medicine, How Luck, Debauchery, and Blunders Gave Rise to Modern Therapeutics. So welcome, Dr. Burwell.
Thank you, Michelle. And thank you to everybody for having me tonight. When deciding what to present on this evening, I could have gone many directions. I could have, taken you through an organic chemistry story of some sort, but I just finished teaching medicinal chemistry. And the stories you're going to hear tonight are stories I've incorporated throughout my lectures in that course. It is kind of the ways I get students kind of interested in medicinal chemistry and drug discovery. And you'll see at the end of the presentation how a few of the recent alums have gone on to be medicinal chemists themselves and some of the awesome stuff that they're doing. So I want to start off my presentation by kind of talking about medicinal chemistry in general and open up the question, does anyone have any accidental medicines they know of before I start my stories? I want to poll the room just to see what you guys know before I start jumping into what I want to say.
Yeah.
Penicillin.
Oh, penicillin. We will chat about penicillin tonight. We will talk about Viagra too. Excellent. You guys must have seen my slides. Well, if you're interested in what I'm going to aspirin, sort of, yes, herbal. We won't be talking about aspirin tonight though. If you're interested in this kind of thing, one book I have my medicinal chemistry students read is the book I have up here on my slide called The Drug Hunters. You don't need to be a chemist for this book. Audible also has it, excellent Audible to listen to if you have time this summer. And it's just about a bunch of stories about the challenges of being a medicinal chemist and where some of the medicines we use today come from. And so when you think of medicinal chemistry, what kind of things do you think a medicinal chemist does on a daily basis? And I have some pictures here that I pulled from the internet. You might think of them as, working with fancy equipment or looking really intently through a microscope, right? Or writing on some sort of glass, all of these chemical reactions that are going to be going on, or, in the lab mixing some crazy chemicals. But the stories I'm going to tell you tonight are kind of where some really common drugs have come from. And they're not from traditional places. So as I go on through the story, if you have anything you want to share, like I said, this is all stories basically tonight. If there's anything you would like to share or know more about, please do not hesitate to interject. I'm happy to just facilitate the conversation, but I have some good stuff in here too. So being a medicinal chemist, I have, dealt with lots of issues associated with medicinal chemistry. And one of the big things is the length of time and how involved the process is. So for those of you that don't know much about where your medicines come from, It's a process that takes almost 10 years from start to finish, usually, right? You would be screening natural compounds. So some of you mentioned willow bark as the precursor to our modern day aspirin. Michelle was talking about using insects for something earlier. So there's a lot of natural sources of drugs that we use today, and that's where they started from. Today, we also get drugs from synthetic chemical libraries. So this is where organic chemists and biochemists put all these chemicals in a bunch of tubes, basically, and then you test the chemicals in those tubes for specific biological activity, whatever that might be. And actually, in combining it with the combinatorial chemistry libraries, Wells students in organic chemistry actually do a combinatorial chemistry lab in the spring semester, where they identify, they synthesize a drug using that approach to identify an antifungal. Of course, this is the age of computers, so a lot of drugs come from molecular modeling and stuff that is not necessarily done in the lab. It's actually done on the computer. And recently, this spring, I taught a biochemical techniques class where students had exposure to some molecular modeling techniques that they can use in the future. And of course, pharmaceutical biotechnology. So you use all of these sources to get a hit or some kind of compound you think is going to be a starting compound. So the stories I'm going to be talking about today is kind of like are the stories of the hit, basically. Once you have a hit, you're always constantly trying to improve it by adding different groups it's like Legos. I tell students a lot like Legos, where you're adding different things to the compound to make it work better. You then have a drug candidate, and then this is in the 2 to 3 year cycle, this is where your preclinical trial data would happen, in vitro studies, animal model studies. And if you have enough data up to this point, you can apply for an IND application so that your drug can enter clinical trials. And then you have a number of clinical trials. Once your drug is approved by the FDA, it is then continually monitored for safety for a number of years. And that's how a drug basically goes from idea to market. Usually this costs billions of dollars for a single drug hitting the market. You might have thousands of hits at the beginning to end up with one drug at the end. So it's you have to be very tenacious as a medicinal chemist, and you can't give up easily. You have to be pretty stubborn to work in this line of work. Okay, so I kind of led with the presentation with this. Have you heard of any accidental medicine? I know some of you have popped up Viagra, which we'll talk about, as well as I forgot what the other one was that I mentioned that we'll be talking about. But I'm going to tell you 5 stories tonight. What I want to let you know is that, and what students walk away from medicinal chemistry with, is that a lot of drugs you take today have a story. And it's not the traditional working hours in the lab coming up with a drug. It's kind of like, oh my God, this makes sense. Or, oh, I didn't think that was going to happen. So a lot of the drugs you take today are drugs that kind of come out of happenstance and accidents. My own PhD drug actually happened that way. There was a molecule that we knew was supposedly protecting mitochondria, but we knew we wanted to add something to it. And so one Friday afternoon, after we'd gotten back from the pub, my PhD boss says, what if we try this? And he threw some stuff in a tube, mixed it up. We gave it to, I studied cardiac ischemia reperfusion injury. We put it on the heart and it was cardioprotective. And so I got to ride the wave of that. It was pretty exciting, with some bumps in the way as well. Okay. So organic chemistry time. Some of you may have heard of these, some of you have heard of these drugs before. So I'm going to test your knowledge of maybe some chemical structures. I've also given you a bit of hints, a few hints here. So I'm going to see, I don't have my chat up, but maybe Rachel, you can tell me what people are saying. The structure right here with the mosquito. Does anyone know what that structure is?
There's nothing in the chat, Lindsay.
In the chat? Okay. So the structure is, we'll talk about it, is quinine. Anybody here gin and tonic fans?
Yeah.
So we're going to talk about
Oh,
Randy piped
just
it in
got
right
it.
when you were starting to say it.
Quinine. So we'll talk about gin and tonics, my favorite beverage. The one with the green growth right here on the mic on the plate. Someone mentioned this earlier.
I think that's
so
penicillin.
it is penicillin. I'm gonna go down here to the blue pill. People know what the blue pill is?
Oh, that's Viagra.
That is Viagra. This one has it on there. Smallpox vaccine. This one could be a little trickier. I have a cow. We'll talk about why that cow is important in a minute.
Anthrax?
Nope, it's not anthrax. It's warfarin. Some of you may know it as Coumadin. Yes. So we'll talk about warfarin and Coumadin and where it came from. And it has a story associated with a cow. Okay. So the first story I want to talk to you about is quinine. And quinine is associated with lots of different things. It is a drug that has done a lot for mankind, actually. It is a treatment, has been found as a treatment for malaria. And I'll talk about that. It is a component of gin and tonic, which I said is very important. And it's also involved in the fashion industry and the color mauve. And we'll talk about why that's important in a minute as well. So let me tell you the story of quinine. So first starting off with the disease. So the disease that we're going to talk about with regards to quinine is malaria. So malaria has plagued mammals and humans for thousands of years. I mean, there was evidence of it in ancient Egypt, but it was probably before that. I would say as soon as humans started congregating, doing agriculture where infectious diseases could flourish, that's probably where malaria developed. Malaria is caused by a parasite called Plasmodium, and it's carried by the mosquito. Okay. The disease originated in Africa, but as people moved out of Africa and traders started trading, it spread across Eurasia, India, and then when European settlers came over to the Americas, malaria found its way over there too. And what you'll notice from this map is that the areas where there are more stringent malarial infections tend to be more in the tropical areas. And so Plasmodium doesn't survive very well in colder climates. This disease actually impacted whole empires. Some claim, and this is totally, like these are all stories, right? That it was actually a main cause of the fall, or a cause of the fall of the Roman Empire. And people in Rome actually would leave the city, the rich people in Rome would leave the city to higher elevations to get away from the malarial outbreaks. What's also really cool, or I shouldn't say cool, but interesting about malaria is that there are certain human health conditions that have developed a resistance to malaria. Does anyone know what they are? Has anyone heard of sickle cell? Oh,
Yeah, thank you.
I was gonna say sickle.
sickle cell? Yep, sickle cell, definitely. And thalassemia. Both sickle cell and thalassemia are mutations in a protein called hemoglobin, and we'll talk about why that's important in a minute. Okay. So this disease is still a major health concern. Medicinal chemists are still looking for therapeutics to treat this disease and in these areas. Okay, so the life cycle of the thing that causes malaria is really complicated, but I like this picture because it's kind of simpler. So what's going to happen is an infected mosquito that has the Plasmodium will bite an individual. The parasite will travel to the liver where it will develop, and eventually that parasite will develop, be released into the bloodstream, and enter the red blood cell. Once it's inside the red blood cell, what it's going to do is that it's going to eat the hemoglobin. So hemoglobin is a protein, which I have here, and this parasite's going to eat this protein. What's really cool or important for hemoglobin is it contains metal centers that carry oxygen for you. So if you didn't have hemoglobin, oxygen couldn't travel around your body. And these little creatures eat hemoglobin. The problem is the metal centers, and free metal centers are very bad. So the Plasmodium has developed a system where it links the metal centers together and kicks it out of the Plasmodiums without hurting the protozoan. Okay. And so when those metal centers are linked together, if you look at the bottom picture of the red blood cells, it's like a grayish picture, you see a red blood cell with a lot of purple dots in it. What those purple dots are those metal centers that are connected together. And so they're called hemozoin. And so those are the things that eventually build up to so much that the cell ruptures. And when the red blood cell ruptures, it's going to release cytokines and lots of different things that are going to cause inflammatory response. And so one thing, if you've watched any pop culture TV about malaria, people that suffer from malaria tend to have fevers that come and go every 3 to 4 days. I don't know. I'm an Outlander fan. If you read the books, there's a person that's infected by Outlander malaria. She has fevers that hit every 3 to 4 days, and that's because of the life cycle of the plasmodium going from the liver to the blood cell and back again. Okay. Now, how quinine works, and I'm not getting all the way into the chemistry on this. How quinine works is it interrupts the ability of the plasmodium to link together those metal centers. So the metal centers become free and kill the plasmodium because they can't link it and kick it out. That's stuck inside of the parasite, which I think is kind of cool. Okay, so there's a lot of folklore associated with the treatment of malaria. And, if someone has heard a different story, please, jump in and interrupt. So malaria was brought to the Americas by Europeans. And what is interesting is probably one of the best early treatments for malaria was quinine. And quinine was a compound that was extracted from the chinquapin tree, which I have shown here. It's also referred to as fever tree. And this fever tree was used by indigenous people of South America people think fever, but specifically for malaria. This tree is native to the Andean foothills. And it was used to treat malaria. So here's a picture of the bark down here where the quinine was actually extracted from. But to remind you, back in the day, a person that was in my field would be considered an apothecary, a medicine man, whatnot, and all kinds of tinctures and concoctions were developed to treat and work and ward off malaria. Everything from garlic to, different types of roots and vegetables and herbs. Okay, just make sure. So how did this come to be? And there's a number of legends. So I've heard two main legends associated with this story. Legend number one is there was an indigenous individual who was suffering from malaria. And prior to that point, indigenous people thought this tree was poison. This person suffered from fever, was very, very thirsty, stumbled to a pool of water, but he was so thirsty, started drinking the water. It tasted bitter, but because he was so thirsty, he did not care. He passed out on the side of the pool of water, woke up, and was malaria free. And then when he looked into the water, the bark from the chinquapin tree had been inside the pool of water. And so they thought that that tree actually had magical powers to cure the malarial disease. The other story I heard was that it was actually a large cat. It was, it was a large native cat that was suffering from malarial disease, because a lot of these diseases were zoonotic, or meaning can affect both humans and animals. And the sick cat drank from the water and then was cured upon waking later after passing out on the side of the pool of water. So I thought that was kind of cool.
Real quick, we have a question in the chat. Rhea, if you want to go ahead and unmute yourself and ask the question, that'd be great.
So you noted earlier that malaria is most prevalent in the tropical climates. So I was wondering from what part of Europe did Europeans bring, because most of Europe is not tropical. From what parts of Europe was malaria brought to the Americas?
So where malaria started was Africa. It came out of Africa and into India. Malaria was prevalent on the salt flats of like Essex. So in parts of Europe, just not as prevalent. But I think
Okay.
most of it was carried over during like slave trade and when they were coming in from Africa as well. It's a good question.
Thank you.
Yeah. Okay. So that was the indigenous people, and by that time, there were Jesuits in South America, and Jesuits learned of the powers of quinine, or powers of the bark, and used it to treat malaria in their own population. So this was around 1630, and it was referred to as Jesuits bark, which you may or may not have heard of before. Once they discovered this, of course, they knew that people in across the seas were also dealing with this malady. So they were the primary exporters of Jesuits bark. This actually was really bad for the chinchua tree because it became over harvested and was hard to find at times, still is. In Europe, though, it was not universally adopted, and you can thank human nature for this. And some of the reasons for why it wasn't universally adopted as a treatment option was due to religious reasons, and I'll get to that in a minute. They were trying to treat other diseases they thought were malaria, but actually weren't. And of course, you got those crazy people out there trying to make a profit off of fake bark. I mean, some things never change, right? With regards to the religious issue, the Jesuits brought it back and people were using it. Individuals in Rome, even the Pope was singing praises of the Jesuits bark at the time. That didn't sit so well with Protestant England. And the Protestants in England viewed Jesuits bark as like a second grade therapeutic. It wasn't good. You know, nobody bought into it. The fact that Oliver Cromwell actually died of malaria because he refused to take Jesuits bark because he didn't think that it was a good medicine. Like I said, some things sometimes don't change, right? But there was a very smart / crafty individual who took advantage of this. His name was Robert Talbor. So Robert Talbor was an individual that traveled all over and made this message. So beware of all palliative cures, and especially that of known by the name of Jesuits powder or Jesuits bark. For I've seen the most dangerous effects followed by taking that medicine. Okay, interesting. Instead, take his elixir and pay him lots of money. Okay. And so this elixir, he sold all over Europe, big names in Europe, Charles II, King of France, cured the King of France's son, malaria, all of these things. And he was actually rewarded quite heftily from the King of France. And the King of France was like, okay, I will pay you 3,000 pounds, which is a lot back in those days, and I will give you a pension that will take care of you for the rest of your life if you tell me the recipe you have in your elixir. And Robert Talbor was no fool. He said, sure, I will tell you what's in my elixir. However, you cannot release the contents until after I die. And the King of France was like, awesome. So he gave the King of France the elixir, the recipe for the elixir. Talbor died, and then it came out the main components of his elixir were opium, wine, and Jesuits bark. And so after that hit Europe, that it was actually Jesuits bark that was in the elixir that cured all these people of malaria, it was then widely accepted all across Europe as the treatment for malaria. Okay, I like that story. Students like that story too. It's pretty good. Okay, so got the British on board. Awesome. So a lot of the British then, would take these to areas that they would then colonize, right? Because the areas that they colonized, India, Africa, some of South America, right? They were places that malaria was prevalent, right? And so the problem with quinine and Jesuits bark was, one, it was expensive and can only be extracted from the chenchua tree at the time. Two, we now know there's some really nasty side effects with quinine, such as bleeding disorders, kidney damage, and heart arrhythmias. And number three, it tasted like crap. So the British fixed number three by basically adding sugar and alcohol. And so that's where you come up with tonic water, and they added gin on the side. So fun party trick is if you take tonic water and expose it to a black light, it actually will fluoresce because of the quinine. Quinine is a compound we call an aromatic compound, which can actually fluoresce in the presence of black light. So fun fact. So this was actually patented in Britain, and actually Schweppes brought tonic water to the US first time in 1953 to sell it. Okay. So like I said, there's some problems with quinine. It tasted like crud. That's what we fixed here. The other thing was that it was expensive. And so after a while, it became evident that people wanted to make synthetic quinine, and people did, but you needed to figure out how to do that. So welcome to medicinal chemistry. So you want to make something cheaper, faster, more available to everybody. So the next person I want to introduce you to is 18-year-old William Perkin. He was a really good chemistry student. He was studying at London's Royal College of Chemistry, and he really wanted to impress his professors so much that he was running experiments at home over Easter break. He wanted to show his professors he could develop synthetic quinine. Well, just like every other chemistry lab, there's some things that don't work so well. And when he tried this, he was trying to synthesize it from coal tar. And he did his chemistry, and at the end of the day, he ended up with a black goo. And this black goo, he thought, oh my gosh, this is just a failure. And so what he did was then threw away the black goo. But as he was washing his hands, he noticed his hands were a brilliant purple. And with the brilliant purple, he got it on his clothes, and the dye the compound would not release from his clothes. So what he did is he invented the first synthetic dye. Prior to that point, dyes were all natural and expensive, and the synthetic dye industry actually opened up the world to make a cheaper, more accessible version of color for people to wear. And the color that he actually developed, the first synthetic color, was mauve. Once he made this dye and he knew he had something, he dropped out of school and set up the first factory to make synthetic dyes. What's interesting is the synthetic dye medicinal world, they are tightly bound together. So there are drugs that have come out as synthetic dyes. I don't know if any of you have gone to the grocery store and seen boxes that are labeled azo for urinary tract infections. Those are actually synthetic dyes. That's actually a drug that came from a synthetic dye. And organic chemists make those synthetic dyes in Wells spring semester. It's one of their favorite labs. Okay, so today quinine has taken the back seat to safer and more effective antimalarials. But however, no one should forget the role this little compound played in medicine, mixology, and fashion. And so here are some synthetic quinine alternatives. And one of these was quite popular last year at the beginning of the COVID situation. Chloroquine, you might have heard of hydroxychloroquine, were both quinine derivatives back in the day. So right now I want to take a reflection. This is my end of my first story. Open it up to anyone that has any comments. And I also want you to reflect on this, because although, I'm a scientist, I was raised by a historian. So reflect on what this little compound did for humankind, and what it meant for the human population for this to be discovered. Think about it in terms of British colonization. If that compound wasn't around, how well could the British colonize around the world? Fashion to the masses, as well as, as one of many, many examples, exploitation of South American ecosystems, right? So this was one example where all of those can hit. Michelle, I see your hand.
I wanted to know if you said that long-time use of it can cause kidney damage and other issues. And so I'm wondering if the colonizers in India, et cetera, who were there for many years, whether they did in fact suffer medical issues from long-term use of quinine.
That's a good question. I don't know the answer to that. I would assume so if they, I don't know the therapy, like how long or anything like
Right.
that. But I
Okay,
will
thanks.
say, well, I will say the tonic water that we consume has a lot less quinine in it, so we don't have to worry about that. You're cool. I did look into that.
They probably had more ill effects from all the gin.
Yeah,
Yeah, that's true.
probably.
That's true.
We've got a few questions in the comments also.
Okay.
Let's see.
Actually, hear an alum downstairs that's coming to my house for a wedding tomorrow. So Belina Mushala is coming to visit. I don't know if any of you know Belina.
So the first question I'm seeing, well, we're seeing a lot of comments about Boo Purple Go Evens. So that's
Okay, no, I get that.
yeah. And then there's one from Renee.
Yeah.
Eric, you want to go ahead, Renee?
Sorry, I heard of quinine for leg cramps.
Oh yes, yes, yes. People have taken quinine for leg cramps. But actually, the FDA has it originally was approved for leg cramps, but the FDA has taken that back just because of the nasty side effects.
Ah, okay. Thanks, Lindsay.
Yep.
So that leads into Ria's question. Ria, do you want to go ahead and unmute?
Yeah, I was just curious to know if quinine is considered to have any medicinal uses currently, or if it's been entirely replaced by the synthetic variants.
For the most part, from what I know, they've predominantly been substituted with the synthetic variants. And so with a drug, right, you can see these structures are very different, right?
Mm-hmm.
And you play with these structures to get them to bind tightly, but not tight enough so that and to get rid of all of those nasty side effects. And they're still tinkering with them. You're never going to get a perfect drug, but they're still tinkering with them today.
And that leads into Sherry, your question about the simple fix.
Well, I just wanted to know, it just seems like if quinine and tonic water is such a seemingly simple fix, why is there still malaria everywhere? Why hasn't it been eradicated? And is this just merely like power and money, that it's...
Excellent question. I get this question all the time from students in the class, right? And it isn't a cure-all. No drug really is a cure-all, except for, we'll talk about the smallpox vaccine. They got pretty lucky with that one. But none of them are the cure-all. And you got to realize that these are parasites. They're constantly evolving, right? They develop resistance. And so I will always be employed as a medicinal chemist because, no drug is perfect. You're just basically playing with what you got at that point in time. And of course, power and money as well. Quinine was really expensive back in the day because before they got the synthetic one, which was late 1700s, it was all the bark that you had to isolate from the tree, which was becoming scarce at the time.
And I think my follow-up with that was, there was a whole episode on MASH.
Mm-hmm.
I know I'm really aging.
I love MASH.
Okay, good. But there was a whole episode on MASH where a bunch of people got sick, and it turned out, I think it was because they had run out of quinine, so they had substituted something else, or they gave them, or the opposite, or I just remember there was something, and then it turned out they said that certain populations couldn't take the medicine. And
Mhm.
Klinger was one of those populations. So he wasn't lying, folks. Anyway, sorry.
Yeah.
But it's like, am I remembering correctly?
So I don't know. I mean, it's been a while since I've watched MASH, but I do know with drugs, and this is getting into a realm of what's known as personalized medicine. What we do know now is as more and more stuff gets known about genetics and what, you know, how much genetics plays a role in disease progression, there are in specific examples, I can't think of one off the top of my head, where certain drugs have nasty interactions with individuals that have certain mutations. On the flip side, right, when I talked about this earlier in the presentation about thalassemia and sickle cell anemia, right, how those individuals were not, to a certain extent, naturally resistant to the malarial infection. And that's because it's a mutation in hemoglobin that causes structural changes in the hemoglobin so that the parasite can't eat it as efficiently and break it down as efficiently. So that's the other, it's the thinking about it the other way, but it goes both ways. Yeah. Any other questions? Okay. I'll move on to my next story. And like, if I start going over, just if you want me to stop, just tell me to stop. Happy to do it. Some other drugs. So the next one I want to talk about is going to be the smallpox vaccine. This therapeutic packed such a huge punch that the WHO, or the World Health Organization, has deemed this disease gone as of 1980. This story, I'm going to warn you, involves some pretty disgusting procedures, cows, and farmers. Okay. So smallpox, like malaria, is a disease that has plagued humans for as long as there have been records, and probably earlier. Probably the first written down evidence of smallpox was in the Ebers Papyrus, which was an early medical text that was kept in Egypt. And it was evident that this disease was present in that population. People that are afflicted by this disease, you know, we've heard about different types of pox, like chicken pox, smallpox. They develop pustules, and in smallpox, the pustules predominantly end up on your extremities and your face, as compared to chicken pox, which tend to be around your torso more. And these pustules, if you're lucky enough to survive this disease, these pustules actually cause severe scarring, and it can actually cause blindness too. Smallpox was a leading cause of blindness as well. Small children are the most sensitive for this disease. And on this slide, you can see the Variola virus, which is shown here, the structure. So this is caused by a virus. Like I said, malaria was caused by a parasite. This is caused by a virus. And the structure of this virus is similar for other types of pox, like chicken pox, monkey pox. There's even horse pox. And, you know, we've deemed this as a puzzle that we've figured out, but there are labs that still have samples of smallpox just in case things need to be studied. You know, we never know how these things are going to evolve over time. So there are people that still have some around, but the last person to actually die of smallpox was I have it on a next slide, late 1970s. So this is actually a really go team for the medicinal chemists. Okay. I tried not to put too many disgusting pictures in here. So there's a quote that I'm going to read at the end, but with smallpox, where the first treatment option for smallpox came from, nobody really knows. They think it may have come from either India or China. And the procedure that was used to treat smallpox was basically taking the pustules of an infected individual and taking the pus from those pustules and scratching an individual that didn't have the disease. And in the process, the individual would get a mild disease, but then they would be protected for the rest of their life from smallpox. It was kind of disgusting, but worked. The process was called variolation, and like I said, there was some issues with it in that some individuals did develop full-fledged smallpox and became scarred and died of the disease. So although it was good, wasn't the best, wasn't great. And there was one individual, so this was done in China, India, and then made it to Constantinople. And if you know anything about the history of medicinal chemistry, during the dark ages and everything, when things were going, when knowledge was going south in Europe, a lot of the medical knowledge actually ended up in Constantinople, and it was kind of a mecca for medical history and knowledge at the time. And so while visiting Constantinople, Lady Mary Montagu came across this technique. And I think she says it the best. So I'm just going to read it. Just the way she describes it. I was reading this. I'm like, this is crazy. So she's trying to convince people to bring it back to Europe. So I'm going to tell you a thing that I'm sure will make you wish yourself here. The smallpox, so fatal and so general among us, is here entirely harmless by the invention of ingrafting, or that variolation that I told you about, which is the term they give it. There is a set of old women who make their business to perform the operation every autumn. The old woman comes with a nutshell full of the matter of the best sort of smallpox and asks what vein you please to have open. She immediately rips open that you offer her with a large needle and puts the vein as much venom as can lie upon the head of her needle. Every year, thousands undergo this operation. There is no example of anyone that has died in it. And you may believe I am well satisfied of the safety of this experiment. I am patriot enough to make the pains to bring this useful invention into fashion in England. And I will not fail to write to some of our doctors very particularly about it, if I knew any one of them that I thought had virtue enough to destroy such a considerable branch of their revenue for the good of mankind. So, first of all, crazy. Second of all, probably would not pass many ethical studies today. But this type of treatment actually dropped the death rate significantly in smallpox in Constantinople. Lady Montagu brought it back to England, and upon bringing it back to England, found a doctor to make sure all her children underwent this procedure. The risk of dying when undergoing this, and what's really cool about smallpox is like one of the first diseases where people actually started keeping track of numbers and understanding whether the treatment was actually effective. And so in keeping track of the numbers, the risk of dying from smallpox after this type of treatment dropped from 1 in 5 to about 1 in 50, which was big. However, like I said, this procedure was still risky, and some people did develop full-fledged smallpox. So people thought there's got to be another way. Cue the cow. Okay, so the virus that causes smallpox are a part of a family called orthopoxes. Inside the orthopox family, you've got cowpox, vaccinia, which is your chickenpox, your monkeypox, and the variola, which causes smallpox. In smallpox, there are two main types. There's major and minor. Okay. What is interesting is not all of these diseases are as lethal as the smallpox. So, for example, on this cow udder that I have shown here, they have drawn in what's known as cowpox. So it's similar to smallpox in that you have pustules form on the cow udder, but however, this version of pox is not as deadly. Farmers would also contract this pox virus from cattle. Once again, another example of zoonosis, where you're crossing between human and animal. But you would get the pustule, they would get the pustules, but they wouldn't die of the disease. It was nowhere near as deadly as smallpox. Okay. So in comes this individual named Edward Jenner. This is the man that was credited with the smallpox vaccine and the first vaccine ever. And the way it was developed, so there was a myth associated with him called the Milkmaid Myth, which I'll get into in a minute. They tried to make his story sound better than it actually was. But Edward Jenner's real story was that he was as a child treated for smallpox using this old technique that I talked about, but became an orphan during that time because his parents didn't survive the smallpox. As he grew, he became a surgeon's apprentice, and during that time became really fascinated in smallpox and treating smallpox. And while working as a surgeon's apprentice, he was working in a clinic that was providing this, crude vaccination, right, with the smallpox pustules. He noticed something interesting. He noticed that certain farmers that came in to get this treatment done on them developed absolutely no symptoms, not even mild symptoms, which most people did when they were given the pustules from the smallpox. Upon questioning the farmers that developed no symptoms, Edward Jenner found out that these farmers actually had just recently dealt with an outbreak of cowpox on their farm. So cowpox had actually protected them from the smallpox. So that was kind of a cool coinky dink. He noticed something was going on, asked the population, figured it out. However, the problem with this is this story got romanticized over time. And just like every good wingman story, he had a friend that wrote his biography. And Edward Jenner was facing some challenges at the time. First of all, cowpox, doing this thing with cowpox, it wasn't a complete cure-all. It had to be done over and over again. Also, people question where he got the idea. Did he steal the idea from somebody? So his wingman decided to romanticize the story and call it the Milkmaid Myth. And his story goes that Edward Jenner figured this whole thing out by basically working with milkmaids and milkmaids that claimed that their beauty came from being immune to the smallpox, because milkmaids never got smallpox. And this is because they were exposed to cowpox. Okay. So once again, this is a kind of a cool little situation where, people just observed it. And it just happened to come of something and was very powerful in fighting a really deadly disease. Okay. So all this came from someone's observation that certain dairy farmers were protected from smallpox. This is the only vaccine that's actually an active virus. So the cowpox was actually an active virus. In some timetables, 1967, the World Health Organization developed a global smallpox eradication program. In 1975, a 3-year-old in Bangladesh was the last person to die of the Variola virus. In 1977, Ali was the last person to die of Variola minor, and to contract it. So 75 and 77 was contract. They didn't die. In 1978, Janet Parker was the last person to die of smallpox. And in 1980, the World Health Organization declared the world free from smallpox. So that is my story about smallpox. Any comments?
Yeah, Lindsay, I can tell you one other thing.
Okay.
The reason smallpox was able to be eradicated were at least two things. One, you could dry the vaccine or dry the virus down. Mm-hmm. In other words, lyophilize it.
Yep.
And you could reconstitute it and it was active. So you could be taken without refrigeration all over the world. Okay. The other is everyone who gets exposed to the virus shows those pocks on their face or limbs. Mm-hmm. So you could definitely tell who had the virus and therefore vaccinate everyone who came in contact with that person. Mm-hmm. There wasn't anyone who was asymptomatic.
Okay. Yeah, and when I was reading about Ali and the child from Bangladesh, they talked about quarantining that individual and they talked about going by foot to everyone. Yeah, exactly what you mentioned.
I'm sorry, Nancy
Nancy?
also has her hands raised, sorry.
Yeah, I have a question, which is about this way of going into the vein.
Mhm.
You talked about, with the smallpox, that they would, I've forgotten the word you used, engraft into the vein.
Yeah.
Does that remind you at all of what the ordinary use of vaccines would, like anyway, vaccines where you just give someone a small amount of it. This is how we used to do vaccination until we got that wonderful mRNA, which is so different.
Yeah.
But before that, we give them a little bit of, whatever the disease is. So would that be at all connected to the way they did it before, where they put it into the vein?
So from what I think you're asking, so yeah, the virus that would have been in the smallpox pustule would have been an attenuated virus that they would in theory have injected. They said vein. I've also heard people call it scratching. Vaccines that are today that aren't the mRNA vaccine, but like influenza, are actually parts of the virus. They're not the whole virus. They're proteins that are part of that virus, so that you develop an immune response to the part of the virus and not the whole virus.
So it's a little bit different.
Yeah, different vaccines have different things. And then of course the mRNA thing, which was new, and being a medicinal chemist in this day and age was really, really interesting. Because that new technology, usually that technology takes years to go from start to finish in the developmental process, but how fast that came about was really interesting. And I was reading about it on a daily basis. It was really cool to see.
Yeah, very exciting.
Yeah.
Lindsay?
Yep.
It's Twitchell. Hi, everybody. There was a really great PBS program on smallpox about 10 days ago.
Oh.
And all of the things that you've been saying were included there. And also, I don't think you've said this, but that Cotton Mather, extremely famous minister in Boston, in 1706 had a slave given to him, such a nice present for a minister from his congregation. One Simus was the slave's name. He was still a slave. He had been given vaccines in Africa. It's sort of maybe in the same way that Lady Montagu had learned about it. And he found out, Cotton Mather asked him about it. Have you ever had smallpox? And he said, I have and I haven't, because I was given this treatment and I got a light case of smallpox and now I'll never get it. Mm-hmm. So Cotton Mather in 1706 tried to convince everybody in Boston to do it, and alienated people, but he was a big proponent of it, and sometimes they used it. So I thought that was fascinating. People did not want to use it because the knowledge of it came from Africa.
Yep.
And they didn't think anything great for medicine would come out of Africa.
Yeah, right. I'm going to have to tell my students about that PBS show. They'll be interested in seeing that.
That was really.
Sorry. My question is, I know that there has been controversy about whether or not to destroy the last remaining stores of the smallpox virus.
Mm-hmm.
I believe they still exist. Those last remaining stores, you just alluded to that. And I'm wondering what your thoughts are about that controversy.
Okay, so Ria, let me just jump in. The only stocks are at Fort Detrick in the United States and in Moscow at Russia. They're very highly controlled.
Yep.
Basically, we have smallpox, parity. Yeah.
Yeah.
I'm glad to hear that there. So, any other thoughts? So it sounds like you're comfortable with keeping those stores.
Yeah, I mean, they're under lock and key. I mean, yeah, I mean, they're there for, maybe not smallpox, but, future, you don't ever know what's going to pop up. like a crazy
Sure.
pot virus can evolve and what can we learn from it, right? But the stores that we do have are, very safely secured. Nancy, did you have a question?
No, I didn't put my hand down. I already asked my question.
Okay. Oops. Okay. So the next story I want to talk to you about is warfarin, or Coumadin. Also involves a cow. But the next two that I'm going to be talking about, both warfarin and Viagra, are a little more closer to home with my individual research that stems all the way back to cardiovascular disease. So start here. It's probably not a surprise to anybody that cardiovascular disease is a leading cause of death in the United States. And when we talk about cardiovascular disease, there's lots of diseases associated with that, such as angina, myocardial infarction, stroke, hypertension, deep vein thrombosis, atrial fibrillation. So there's lots of different specific conditions that are lumped under the term cardiovascular disease. It can also be caused by genetics. So, my dad specifically had valve abnormalities where he had to have heart valve replacement, right? So there's lots of things when we talk about cardiovascular disease that fall under that umbrella. So this is a map from the CDC that I got just today, the most updated map they have from 2014 to 2016. The more red means the disease death rates associated with heart disease, and showing the map here. Okay. And so when we're talking about heart disease, we talk about, like I said, heart failure, you have aorta disease, you have coronary heart disease, you have risk factors that contribute to heart disease, vascular disease, arrhythmia, heart valves, cardiomyopathy. So there's lots of different terms that get thrown around with regards to heart disease. And some treatment options for these include angioplasty, where you go in and, for example, if you had a blocked vessel, you could open up the vessel using angioplasty. Pacemaker is if you have a heartbeat that's irregular. Pacemaker might be used to make the heartbeat more regular. Valve transplant, so if you have a hole or a rip in the valve, heart valve, it can be transplanted, as well as hypertension medication. And warfarin or Coumadin. So anyone that's being diagnosed with one of these heart diseases or has had a certain type of surgery probably would also be prescribed some kind of blood thinner. And warfarin Coumadin is one such blood thinner. But where did it come from? And the answer here is bovines. So the story for this drug goes back to the 1920s. And in the 1920s, cattle in the northern US and Canada were afflicted by a disease that resulted from fatal bleeding, either spontaneously or from a minor injury. And what was unusual is particularly seen in 1925, because in that area that year was extremely wet year and super economic hardship. We're getting close to the Great Depression. Farmers, I was a dairy farm kid, so, times were tough. Times are always tough on a farm, but they were even tougher then, apparently. And so, making sure nothing went to waste, definitely. Okay, so these cows were sick. And so an individual that tried to figure out what was going on was Dr. Frank Shofield. And Dr. Frank Shofield was a Canadian vet. He graduated from Guelph University. And he was the first one to figure out what was going on with these cows. He found out that the cows were actually sick. The cows that were sick had actually consumed moldy silage. I don't know if any of you are farm people, but if you've ever smelt silage, it's horrible. And moldy silage, even worse. So contained moldy silage that contained a plant called sweet clover. And I have sweet clover shown right here. Like I said, the majority of these cases were between 1920, or was around 1925. And they were dealing with some pretty big economic hardship. So they were trying to use every last bit of feed they possibly could for their cattle. Hence the reason they, feed that, may have looked slightly okay, but maybe a little moldy, they may have, mistakenly used. This condition, because of the correlation with sweet clover, was termed sweet clover disease. And treatment, if you were lucky that your cow didn't die, it was changing feed or giving it a blood transfusion. It was also considered at this point, remember, the chemical was not known at this time. It was considered dietary deficiency. Okay, so that's basically where it ended, and cows were getting sick. Farmers would call the vet. The vet's like, you just got to change your feed, and it's sweet clover disease, and it's some sort of dietary deficiency, until Ed Carlson. Ed Carlson was a farmer that had many of his cattle die. He consulted a vet, and it was told that he had sweet clover fever, and it was caused by a dietary deficiency. He was told the only treatment was a transfusion and changing the cattle's diet, just like I could see my dad doing this. Ed called BS on the veterinarian and drove a dead heifer, 100 pounds of moldy silage, and a milk can containing uncoagulated blood through a blizzard to the University of Wisconsin. Okay. By the time he got there, everyone's lab was shut down. He found an open lab. The biochemist in the lab was Carl Link. He went in and said, Carl, you need to figure out what's going on. Here's the blood, here's the silage, here's the cow. Use whatever you need to figure out what's going on. And within 6 years, Carl had identified the compound dicumarol as the anticoagulant that inhibits the blood clotting cascade. And from there, he ended up developing warfarin and other anticoagulants by this happen chance crazy farmer that just wandered in his lab one day. So dicumarol is actually a compound that is produced by the mold that grows on sweet clover. Coumarin is synthesized by the clover that is converted to the dicumarol. Carl Link was an eccentric dude, so this doesn't where it ends. Carl Link was a pretty eccentric dude. He figured out what was going on with the cattle. But he suffered from bouts of tuberculosis. So while he was recovering from a bout of tuberculosis, he had this amazing, brilliant idea. He's like, you know what I can use this compound for? I can make lots of money out of it. I can use it as a rat poison. And so this compound was used initially as a rat poison. Now, okay. So up until this point, warfarin was predominantly used as a rat poison. There were ideas floating around that people might be able to use it for different conditions that might promote clotting, but they, without an actual antidote to it, they didn't want individuals to bleed out or hemorrhage, right? So it was considered really dangerous. It was a rat poison for sake, right? And so they didn't want to give humans rat poison. That's just, ridiculous. So using it as a human therapeutic didn't really happen right away. What caused it to be used as a human therapeutic was after an individual, a soldier, had just enlisted into the military. He didn't think that was a great idea, so unfortunately tried to think he was going to try and commit suicide by consuming a huge amount of warfarin, estimated to kill an adult male. Now, what he didn't know is warfarin doesn't work instantaneously or like that. And after 4 days, he came to the realization he wanted to live. He walked into an emergency room with a bloody nose. The medical professionals that were on his case were left to their own devices to think quickly, oh my gosh, what do I do? And so I'm going to go through here and talk to you about how warfarin works. And then I'm going to ask you, what would you do if you were in the ER and this person came and you had to treat this person? So how warfarin works, in the normal blood clotting cascade, there is a version of vitamin K called reduced vitamin K. And in the presence of carboxylase, it takes precursor thrombin to biologically active prothrombin. This prothrombin is needed to clot. And in the process, you go from reduced vitamin K to oxidized vitamin K. Now, in the body, vitamin K is a continuous circle, cyclic, right? And so you can take that oxidized vitamin K and recycle it back to reduced vitamin K. The problem is with warfarin, what it does is it inhibits that recycling process. So without the reduced vitamin K, you're not making the biologically active prothrombin to clot. So I'm going to ask you, if you were his doctor, what would you do? Or what would you think would be a treatment that you could use to save the soldier? Any ideas? You can give him anything. You have the whole chemistry cabinet at your will.
Sherry, go ahead.
Well, you would give him potassium, vitamin K, give him spinach, give him kale, things like that. That's what I have to do when my Coumadin level's high.
Oh, okay. Yes, you would give them. So the individual ended up giving them reduced vitamin K. And what was really interesting about this specific situation, it was the first time people have shown that you could save somebody who overdosed on the blood thinning medication by modulating it with the vitamin K. And so then it was then used as a therapeutic. It's probably one of the most widely used therapeutics, associated with cardiovascular disease. My dad's on it, when he had his valve transplant, he will always be on it, always constantly checking his INR, or his ability of his blood to clot. And so that's basically how warfarin came to be used as a therapeutic for cardiovascular disease. Any questions on that one? And Rachel, tell me or Emily, tell me if there are no questions.
I don't see any questions at this time.
Okay, because I know you're all waiting for the next one. So the next one I'm going to talk about is Viagra. And Viagra actually was patented for an unintended use. So students love this story. Sildenafil was its actual scientific name. Viagra was an interesting drug that made Pfizer a lot of money. No one had ever invented a drug to treat this condition that Viagra ended up treating ever before. It was the only one in the market that had a drug that did it. It was an FDA approved for treating erectile dysfunction, which was not its intended use. The revenue for Pfizer has dropped in recent years because last year this drug's patent expired. So if you're in the drug discovery business, if you patent a drug, the drug is usually good for about 20 years, and then it goes off patent where you can start making generics. So it ended up going off its patent in 2020. It hit the market in 1998. It's used by over 62 million men all over the world. And total revenue in 2019 was down actually to previous years to 500 million because it was starting to come off of its patent. So really crazy drug that happened by accident. So vascular tone, or the tone of your blood vessels, is really important for heart health. And under pathological conditions, you may have heard the term hypertension. What happens in hypertension is the vessels in your body basically constrict, and that's caused by lots of different things. But that constriction actually is really bad for your heart because that makes your heart have to work harder. And when your heart works harder, if this is done over a long period of time, can lead to conditions like a cardiac hypertrophy, where the muscle around the heart actually increases, and that can cause problems with the chamber. It won't fill as easily. And so really, really bad stuff can happen. Okay. Pharmacology is really important in treating hypertension, and there are multiple ways to treat hypertension. I'm going to talk about a couple ways here, but there are lots of different ways. I have a whole class just on hypertension and the pharmacology associated with it. The first one I want to talk about, and if you ask any of my students, they'll tell you how much I'm obsessed with nitric oxide. Are the vasodilators, the nitric oxide vasodilators, and one in specific called nitroglycerin. This is still used for hypertension. How this works is this is what's referred to as a nitrate molecule. All that means is it's going to go inside the vessel walls and eventually will release this compound called nitric oxide. Nitric oxide will activate an enzyme that'll eventually produce this molecule here called cyclic GMP. And cyclic GMP is the thing that tells the muscle around the vessel to relax. When it relaxes, it opens up the vessel, allowing blood flow to come through more efficiently. Okay. So fun fact about nitroglycerin is that before being used as a therapeutic to treat hypertension, Alfred Nobel, yeah, that guy, Nobel Prize guy, he worked with this compound in the explosive industry. He made a lot of money off of nitroglycerin, but by the end of his life felt extremely guilty with all of the damage and hurt that he caused by developing such a powerful explosive. And that's actually why he set up the Nobel Prize, was because of all the guilt he felt. And it didn't stop there. He was so turned off by nitroglycerin that when he was diagnosed with angina later in life, he refused to take nitroglycerin as a treatment and eventually died of heart failure. So the thing that he invented could have helped him, but he didn't take it. Just a side story. Okay. But back to Viagra. So I love this story. This is like the typical science story. So the individual that came up with Viagra was David Brown. He was a Pfizer chemist. And in the early 1990s, in front of a clinical development committee, he got crucified for wasting money. He was developing this compound to treat hypertension. It wasn't working. He wasn't getting the data he needed. And they were telling him that this was just a ridiculous idea, and told him to come back in September. And if he didn't get any additional good data, the project was just going to get shut down. That's the way drug companies work. They have X amount of time. You need to produce results. And if you don't produce results, they're just going to kick you out. Okay. Like literally days after that meeting, he had a study set up with Welch miners. He was looking for anything he could report on with this drug. He was wanting to look at hypertension, but anything. And at the end of the day, he wasn't seeing anything too extensive. So he just decided to ask them, like, how do you feel? Are there any weird side effects? What are you seeing with this drug? And one miner sheepishly blushed. And he said, well, after taking this drug, I tend to have an erection. And then all the other miners turned around and said, we did too. And so David Brown's like, what? And he went back to Pfizer's clinical development group. He stormed into the office and asked for 150,000 pounds for an impotence study. The group said initially no. And David Brown said, I'm not leaving your office until you give me the money. And so by the end of the day, they did give him his money. They did agree for him to do the study. They used it during the study. They measured the effect by using this thing called a Rigiscan, and basically found the only drug, right, that was used to treat that condition. And that's why Pfizer made a lot of money on that. Okay, so how does this work compared to the nitric oxide that I told you about? So in this condition, right, you're also looking for vasodilation. And why was it the specific piece of male anatomy that became affected after the treatment? So what the drug actually does, so I told you about nitric oxide making cyclic GMP, and cyclic GMP leads to relaxation, or in this case, an erection. Well, what the drug did was it was an inhibitor for an enzyme PDE5, so phosphodiesterase isoform 5, that is used to break down cyclic GMP to turn the signal off. So if that was inhibited, cyclic GMP stayed around longer, and led to physiological effect. The fun fact no one realized was phosphodiesterase isoform 5 was significantly found more predominantly in a certain piece of male anatomy than other areas of the body. Hence, Viagra was discovered. Oh, but it didn't stop there. Finally, they found what they were looking for. They had enough data. They did the epidemiology. They did find that the drug Viagra, or the actual chemical name, sildenafil, did have an antihypertensive property and was used and approved by the FDA to treat pulmonary arterial hypertension. This was approved by the FDA to do this in 2005. The only thing that's different between this drug and Viagra is the dose. So this dose is 20 milligram tablet, where Viagra tends to be 50 or 100 milligrams. It's the only difference. That is my Viagra story. Any questions on that one?
No, just some comments.
That's okay. I knew I was gonna get that. That's the debauchery part, you know?
Yep.
I had to put that in there. Okay, so I have one more story.
Oh, I was just going to say, Kristen Schaff is the reunion vice president from the WCA, and she said you would have turned me into a science major if you were at Wells when I was. Rhea Hirschman actually has a question.
Yep. I hear Professor Elliott's in my house now too, apparently. Okay.
There we go. I'm wondering if you know about a documentary called Orgasm Incorporated.
No.
It's a very good film, which is a little bit tangential to what you're talking about, but it's related. It's about the pharmaceutical industry's desperate attempt to create a parallel drug for women based on virtually no scientific evidence, but a lot of marketing and a lot of playing on women's insecurities. So it was made about 10 years ago. So some of the statistical information is a little bit off. But for instance, in there were these claims by various pharmaceutical companies, that supposedly by surveys, that 43% of American women suffer from female sexual dysfunction. Without actually ever defining what that means. And it's actually, so I highly recommend this.
Yeah.
It's a little bit, again, it's a little bit off from your main topic, but there is what might be interesting is the way in which the pharmaceutical companies manipulate what little science there is.
Yeah. Oh, yeah.
In an attempt, as I said, to create a parallel drug, even though the biological functions are actually very different.
Right. Yeah, that would be really interesting.
Yeah, so I'll just type the name of the video in here, and I highly recommend it. I use it in some of my classes. There you go.
Thanks. Okay. So the last one that I'm going to talk about, my last story, is penicillin, and I left that one for last just because this is kind of how I'm feeling at this moment with the end of the semester. Okay. So before the introduction of antibiotics, there was no effective treatment for bacterial infections such as pneumonia, tuberculosis, gonorrhea, rheumatic fever. Also, many soldiers died in wars that were not due to be killed in combat, but rather infections that ran rampant on the battlefield. Penicillin was the thing that changed all that, and was discovered in 1928. And by the time World War II rolled around, it was produced in mass quantities and for soldiers on the battlefield. As more understood regarding what penicillin could and couldn't do, more antibiotics were developed. So different antibiotics have different mechanisms of action. I'm going to geek out a little bit and talk about the chemistry of how penicillin works, but they all have their own different mechanism of action. However, none of these would have been discovered unless there was a messy biochemist who decided to leave for summer holiday without cleaning his bench. Okay. So this individual was named Alexander Fleming, and he was a biochemist that was working on influenza. And in the labs, in the research facility he was in, he did have the bad reputation of being one of the messiest technicians in the building. He was working on a study focused on influenza when he was ready to leave for summer break. However, before break, he didn't do what every good, microbiologist, biochemist should do in, cleaning out their plates, putting their plates away in the fridge so things don't grow on them. When I say plate, what a plate is, right, is basically kind of like a firm jello where things can grow. We have lots of sugar and protein, so lots of bugs and creatures can grow on them. And he left it out on his bench. And when he got back, he noticed the bacteria, Staphylococcus bacteria, and mold on his plate. And what was interesting and what he noticed about that is no bacteria at all grew around the mold. He went on to identify the chemical in the mold that prevented the bacteria from growing, and that chemical was penicillin. So I like this little quote at the bottom, right? It kind of sums up the presentation from today, I think. When I woke up just after dawn on September 28th, 1928, I certainly didn't plan to revolutionize all of medicine by discovering the world's first antibiotic or bacterial killer, but I guess that was exactly what I did. Alexander Fleming. As time went on and they were looking at how to use this antibiotic, in 1941, Alexander Fleming actually received the first dose of penicillin for a life-threatening infection. However, unfortunately, they didn't make enough dose, and so he died before seeing the impact of antibiotics on the medical community because he didn't receive enough of it. A year later, the drug was used by Ann Miller, who was treated for an infection after a miscarriage and was successfully treated with antibiotic. It was used extensively in World War II. And just kind of talking about the punch that this therapeutic packed, death rate from bacterial pneumonia dropped from 18% to 1%. And in 1945, Fleming won the Nobel Prize in medicine for his discovery. Okay, so I'm going to geek out. I only have a few slides on this, promise. So I kind of want to talk to you about how penicillin works. So penicillin doesn't work on all bacteria. It only works on bacteria that has what's known as a proteoglycan, or I guess on this slide it says peptidoglycan, outer layer. All this is a number of layers of amino acid type layers that are stuck together. These type of bacteria are referred to what's known as gram-positive bacteria. And when you do a stain, you see these little purple things. Those are gram-positive bacteria. As contrast to gram-negative bacteria that don't have that wall and therefore aren't stained as efficiently. So penicillin works on gram-positive bacteria. And you might ask me why? How does that work? And so how this works is there is an enzyme, the little machines that do everything inside the cell, that basically put those proteoglycans together. They attach amino acids together so that you get the multiple layers. Well, how penicillin works is it binds to that enzyme and won't let the layers form properly. And when the layers don't form properly, what happens is water that enters the cell, it no longer has a rigid structure on the outside. And so water bursts the cell because it doesn't have the structure to keep it in anymore, and therefore the bacteria is killed. Okay. So this evening, I've told you a story of 5 therapeutics that were kind of found by accident. Had some big punches in humankind, in human history. But these are not the only ones. I could do a whole course on accidental medicine. Things like you talk about angiography when you have like a clot in your heart, right? And you want to image where that clot is. That technique, that therapeutic technique or that diagnostic technique was actually found by accident. Some technician accidentally put the contrast material into the vasculature in the heart, which they thought was a big no-no, but then ended up being one of the best diagnostic techniques to image heart vessels. So there's lots of stories on where your medicines come from and how and diagnostics come from that may scare you a little bit, but it's kind of a cool story. Each medicine you take has a cool story associated with it. And so where I want to leave you is, so I've now taught medicinal chemistry for 3 years at Wells College. A number of alums have gone through that class. The one at the top's in my house right now, and are now their own drug hunters, medicinal chemists. So Belina Mushala is getting her PhD in pharmacology and chemical biology, just qualified. So she's now a PhD candidate. And I believe has 2 publications out now, and she's only been there. You can see when she graduated Wells, powerhouse. Al Milliken actually is doing his PhD in my PhD lab with my PhD advisor. It's a little weird, but cool. He just got an AHA grant. He works on cardiovascular disease. Liam Kaler, he's at University of Pennsylvania. He is working on COVID, actually, and looking at binding proteins for COVID. Molly Waltz just graduated a few weeks ago. She's actually going to become a pharmacist, going to the University of Binghamton. And Lexi Cohen let me know that she just graduated with her PharmD from Temple University to be a pharmacist. So there's a lot of students that are interested in pharmacology, therapeutics, and medicinal chemistry. And I'm really excited to see where these people end up in future years. And with that, I kind of want to leave it there. Thank you. And I'll take any questions, comments, whatever you may want, whatever.
Lots of awesome comments. So definitely want to read through those. Thank you so much. Oh, Ria's got a question.
Put you on mute.
Okay, so the discovery of penicillin, as we know, it was an accident, but was there, I've always wondered about this actually, was there an active pursuit of what we now call antibiotics at the time that Fleming made this accidental discovery, or was this just something that to everybody seemed to come totally out of nowhere?
Oh, good question.
That's a good question. I don't know the answer. I don't know the answer to it. Like I said, this is a little out of my wheelhouse with regards to like where my niche is with cardiovascular disease. I put it in there because I figured, I mean, that's just one I've always heard as an accidental medicine, but it could be myth. When I was digging in Edward Jenner's story about the Milkmaid, right? I've always been told the Milkmaid story, but when I was digging and doing research for this presentation, I found that that was actually a myth. So I'd be interested in looking at Fleming to see. And I know other areas of antibiotic research, like targeting, for example, ribosomes and things like that, have been actively pursued, and they weren't just accidents. So I'd be curious to see where that fits in and dig a little more on it.
You can keep going if you want to.
I was going to say there are lots of nice compliments, and Deb said this was much more interesting than the freshman chem classes I took at Wells. Congrats on your promotion. I thought that was really nice because science scares people sometimes, and you know, if it's interesting, it's engaging.
I can roll out the organic chemistry. You guys want to do Fisher esterification? Let's go.
You know what? Let's stay on. Let's get wild. It's reunion.
Yeah, right.
Right. That's right. That's right.
If I did this in person, I think we would all have had to have gin and tonics. I'm just saying.
Yeah. Well, I was saying, I'm like, I guess that's like our new reunion drink. Randy was really all about it. I support that.
I stepped away to make one, and I came back, and you were talking about Fever Tree, which is in my drink. It is Thursday night during COVID. It's a new tradition here, so.
Mine's downstairs. After I'm done, I'm gonna have one.
Okay, so I have a question, and I know nothing, but I was just reading and hearing, I don't know, in the past couple days, and you may have mentioned it when I stepped out to get my Fever Tree, but I'm really fascinated by this idea, and maybe it's your next lecture, that we could cure cancer by using a similar kind of vaccine method, which is you don't just inject a little bit of the virus, but you get the body to mimic the protein, whatever it is, to trigger the immune system. Is that stuff science fiction, or is that
is stuff that is actually that is stuff in preclinical trials, and that is stuff you actually should ask Liam Kaler about, because I think that's part, I mean, he's working on COVID stuff, but his lab is focused on cancer type research. Probably could answer it much more, or a lot better than I can, but I will say that it is preclinical trials. I have read about it. I haven't had 2 seconds to dig into it because, like you said, it's fairly new. It's fairly new. That's been out in the last, I would say, before like this last semester came out, or it came to my attention anyway.
It just seems so cool because the idea of having a little bit of cancer injected into you is not really
Yeah.
the thing that would come to mind, but if it could get your body to think it needs to, yeah, that's
Right.
really cool.
Yeah. A lot of science ends up like, I remember as a little kid watching things like Star Trek and things like that and be like, oh, that'll never happen. And then you see how some of the stuff develops. So like when I was a grad student, I was at the time, they were looking at stem cells, and we did what's called the Langendorf Perfusion, where we perfuse the heart and can do heart attack and things like that. But people at other places were basically taking the heart muscle on and off the vessels and then putting and perfusing with stem cells, and new heart muscle would form and the heart would beat. So completely regenerating the heart. I don't know where that research went after that. Obviously, I went to go on to Cornell and do genetics and teaching and such. But some of this stuff is rooted, like you read it in science fiction, you're just like, oh my gosh.
Oh, I learned all that watching like 25 seasons of Grey's Anatomy. So.
Oh, okay. Got it.
Courtney, why don't you go ahead and ask your question? Sorry, marketing major over here.
And then we also have Julissa, who has a question after Courtney.
Hi, this is Courtney Jones. So I'm a biochemist and have done a bunch of my research on metabolomics and natural product research.
Nice.
So you're on, talked a little bit more about like the synthesis side of things. But I was wondering if you could share your thoughts on, kind of current pursuits of new drugs in terms of the natural product side of things, and especially in regards to kind of problems within that type of research, especially like rediscovery.
Oh, like where do I start? And finding new uses and recycling things, because that's one thing with drug discovery is if you can find a way to use something that's already been approved for something else, that decreases the time and effort you need to go to do a lot of the approval research for the FDA. Did you have a specific one in mind?
Not in particular.
I could go down a rabbit hole and talk about metabolomics, because I love talking about metabolomics. When I was at Cornell, I decided to do a project on erythritol, in nutrition. And we were on a study looking for metabolites that could predict whether the freshmen gained 15 pounds or not their first year, that freshman 15. And with the thought behind that is if we could find the metabolites that gave us a trigger that, maybe they'd gain the weight, we could intervene with diet so that they would have better health outcomes later in life. And this study's still going on. It was the last study I did at Cornell. And when I did it, there were a couple metabolites that popped up, branched chain amino acids, which tend to pop up when someone's undergoing, metabolic stress. But one was unusual, and it was erythritol. And erythritol is a sugar that's naturally found in plants. It's made by plants. Originally thought to be made by plants. And it's found in chewing gum. And so I was working in a group of epidemiologists, and they're like, okay, we're ready to publish our paper. We got our results. It's erythritol and these branched chain amino acids. And the biochemist, me in the group, the only one, was like, that's weird. We need to look into that. And so, and this was done with Pat Casano. She's the director of nutrition at Cornell. And she's like, okay, let's see. And so she talked to some of her colleagues, and we did a study where I actually got to bleed some of my colleagues the last day I was at Cornell. I collected blood, or we had a phlebotomist collect the blood. After we gave them labeled glucose and asked the question, where does the glucose go? And we collected the blood and did analysis, and we found that humans actually can make erythritol. And that ended up being published in PNAS. And since then, a lot of people have gone into work. I have worked on individuals with metabolic syndrome, the enzymes that involved in the pentose phosphate pathway, how are they altered with people with metabolic syndrome, and that erythritol is produced in people that are that have a propensity of developing metabolic syndrome. So it might be an early marker for individuals that have that condition. And of course that ties into cardiovascular disease and everything like that. But that's my story of the natural product I was working with in metabolomics.
Very cool.
Yeah.
If Julissa or Sherry want to go ahead and chime in, and then I think those will be the last two questions and we'll wrap it up.
Hi, this is Julissa. I wasn't sure if I missed something while we were chatting regarding like the cancer treatments. I had recently read that they were seeing that the messenger RNAs may be effective, like that type of vaccine toward treatment for cancers, and a couple other things. Is that what was in clinical trials, or was that something different?
So I'm going to be completely honest, like when I first came across it, what I think Joanne was talking about this semester, I haven't had time to delve into all of that research during the semester. I just knew that it was on the radar. I don't even know about the mechanisms. That's something I'd like to look into in the future. Being a cardiovascular biochemist, I mean, I get bits and pieces from the cancer world, but I have to have time to get that when that gets on my radar.
Sherry, last question.
Yeah, mine was just a question about the penicillin. I know when I was little, I caught spinal meningitis, and at that time they said the only thing they could give me was penicillin. And I was allergic to penicillin, so this was like a big deal at the hospital. But now I think that there's like a lot of different, are there like a ton of different antibiotics that if you had meningitis, you could, like, did they just figure this out with different molds, or are these synthetic things, or?
Yeah. So penicillin actually isn't used all, like, just straight up penicillin isn't used all that much today because of resistance. So bacteria develop resistance to the antibiotics. So there's constantly new antibiotics that are being formed and identified. If you read the Drug Hunters book, at the beginning of the book, I think they actually talk about isolating antibiotics from soil. And I know Professor Elliott teaches genetics at Wells. She does a lab with that in microbiology, like looking at the quest for antibiotics out of soil samples. So there's lots of sources for antibiotics, and that's constantly something that's always being developed.
They also have like vaccines now, they said, for meningitis and chicken pox and stuff that we didn't have when I was little. And like when you were talking about the smallpox being close to chicken pox, and it was just like, is that what made the chickenpox varicella vaccine? Like, did they build on smallpox stuff, or?
I don't know.
Is that how it works?
I don't know the actual process they took in developing the chickenpox vaccine. They could have looked at past research on I think I could be wrong. I'm not a virologist by any means. I could be wrong, but I believe that was, like I said, part of the chickenpox vaccine or chickenpox virus. So smallpox was a live virus that was used to the cowpox. It was a live virus that was used to treat smallpox. The chickenpox vaccine was, I don't believe, live. I think it was either attenuated, dead, or a piece of the virus. So like I said, I don't know the story behind how the chickenpox vaccine was developed, but I would guess one of those ways.
Okay, I'm sorry. It's like, I don't know. This is not my field.
No, that's okay. No, and what's really cool, right, is with the COVID vaccine, which has happened like this year, was a totally new way of doing vaccines with the mRNA. which isn't even a bit of the old virus, right? It's the mRNA. So what mRNA is used for is you have a gene, right? A gene encodes a protein, but before it can go to a protein, it needs to be converted to mRNA first. So it goes gene, mRNA, protein, and then it can do its cellular function. Previous vaccines used pieces of protein or pieces of virus, but what the COVID vaccine did, it was use the mRNA that was encoding for one of the proteins. So it's a completely new technology for this specific virus.
If we didn't have COVID, do you have any idea which like disease they would have used this new technology for next? I mean, like, was that already in plan for something else and then they just switched lanes?
From what I was reading, they were working on it. Just because COVID happened, it was fast tracked so that it could be, go through that FDA process faster. So they were working on it. I don't know for what virus they were working on it for, but that was something that was all that was being worked on. That's one of the reasons it also came fast. It just didn't come out of nowhere.
Yeah, because everybody was kind of like, well, how did they suddenly know how to do this?
Yeah. No, it had been a thought in the back. It had been a thought.
That's good.
Well, I definitely, love and appreciate the conversation, and Lindsay, so much for your expertise. Like, this was awesome, and I know people really enjoyed it. Thank you for all the amazing questions. But I definitely want to be mindful of everyone's time, and that Lindsay has alum guests in her house right now, which is very exciting. So I hope everyone has a great night, and thank you so much for attending. I'll get the recording on the YouTube page as soon as I can, so you can watch it again and maybe pick up on something you didn't catch tonight. So thank you so much, everyone. Have an awesome evening.
Yep. Feel free to email me if you have any other questions.
Thanks a lot, Lindsay.
Yes, I'm going to pop my email in the chat real quick in case anyone needs a link for other reunion events this weekend and you don't have one. Just send me an email at rporter@wells and I will send you the link. Have a great night. Thanks, everybody. Thanks, Lindsay, Professor Burwell. We appreciate it.
Thank you.
Bye.